Okay. Boop. All right. Cool. So welcome to my talk about some really fun things that we've been doing on the whole LA watershed, not just the LA River. So this is a joint crash space UC Santa Cruz project. So environmental DNA in the Los Angeles watershed, how to turn 100 gigabytes into a few bullet points. Actually, at the end of all this, it's probably even more than 100 gigabytes of data. So, all right. Okay, overview time. I learned so much about giving talks. So first off, LA does have a river. Second is that I'm going to get into some of the background behind environmental DNA or eDNA for those in the know. And then getting into remote sensing. So looking at the environment from space. And then after that, like, what does any of this mean since we're collecting all this data? And that's going to lead into the last part of this talk, which is also going to lead into the second talk I'm going to give later this year. So it's all part of the eDNA cinematic universe where we lead. It's all going to wrap up in the molecular ecology endgame, which is a 3-hour talk I'll give. And then so part 2 of this is like, what does any of this mean? Now what? And then where we're going in terms of managing eDNA data and like big data ecological data sets, and like how the LA watershed project is sort of a test case for a bunch of things we're building. All right, so yes, LA does have a river. Look at that river. That stretch there, sort of the Glendale Narrows, to me is the most picturesque part of the river. But yeah, it is 51. The LA River proper is 51 miles. It is navigable. It takes 2 days on a kayak if you're really determined. And the watershed itself, is highlighted there, has a bunch of tributaries. One of the ones we actually are most interested for this project is the Arroyo Seco, just to the east there of Glendale. And anyway, so it drains a pretty large area, a very heavily urbanized area. And anyway, when you think of the LA River, you're probably thinking that it's, yeah. So you're not usually thinking of something like the Mississippi or the Hudson or like a bunch of other river systems. You're thinking something that's more of like a natural habitat for shopping cart breeding. So these are some shopping carts in a native habitat. You're also thinking about it's a really great location for lots of famous movie shoots. So Gone in 60 Seconds, Grease, and of course, the most famous movie of them all, The Rollerblade 7, directed by Scott Shaw. I highly recommend you look up Zen filmmaking if you want to ruin your life. Okay. Oh, yeah, by the way, stop me at any point if you have questions. I don't really care about waiting until the end. If something does not make sense, say something.
I got a quick one, Levi.
Yeah.
So if you're in Santa Monica and you're driving back and you cross over a thing and it looks like a river, but based on your thing, it's not the LA River.
Yep.
Which river is that?
That is the Bayona Creek. So, which is actually a separate watershed than the LA watershed. So the Bayona Creek, drains on that's the like, Marina Del Rey area, Culver City. However, historically, the LA River, by the way, the reason why the LA River takes this course, this back here, the reason it takes this course is that we, and by we, I mean the Army Corps engineers in the first half of the 20th century, completely concretized the entire system to prevent it from changing directions, because before Europeans got here, actually really, I mean, before the Army Corps engineers, is like naturally this river, because it's a very, very flat drainage system, is it changes direction, like where it goes every 20 to 30 years. So like the Tongva knew about once generation, the river is just going to pick up and like go somewhere else. And you just kind of have to go with that, because like, what are you going to do about it? And it would sweep anywhere between about Seal Beach, which is like to the southeast of Long Beach, all the way out to close to the Bayona wetlands. So it's a huge area that it would sweep through, and it would change directions in the part south of Griffith Park. Once you get into the San Fernando Valley, it's more naturally channelized. But yeah, it'd sweep through a very large area. So, but yeah, we concretized it so that it wouldn't do that. Disobedient river. Okay. So also, which is always surprising to people, the thing about with LA, especially if you've grown up here, is that we have this river system, and this river system happens to be in one of the 36 global biodiversity hotspots on planet Earth. So the Southern California something floristic province. I'm totally an ecologist, but anyway, this is actually a good map at 1 kilometer resolution of what's known as the potential biomes of the Los Angeles area, which is our best guess given a lot of historic data, climatic data, and ecology, what biomes would exist in Southern California or the Los Angeles area without any human modification. I mean, this is sort of our best, this is a scientific best guess. But yeah, like we actually happen to live in one of the biodiversity hotspots on planet Earth, even though we're also the second largest urban agglomeration in the United States. We're not unique globally in that sense. There are other cities that house 70 in biodiversity hotspots. Singapore is a very famous example. But this like leads to like why we're interested in studying the ecosystems in Los Angeles. LA has a bunch of like very rich ecosystems. Of course, and the question people always ask is like, why would you study this? Like when they think of LA or they think of a lot of urban environments, they think of pigeons, coyotes, and rats and other things like that. But it turns out these are very complex ecosystems, and they're a result of human activity. So if you were to zoom way out, you would actually look at human beings no different than you would with coral polyps. We build cities to live in, and then everything else will like grow on top of it. So the strange thing is because of human activity in Los Angeles, so it's in effect we terraformed this place. There's more biomass today in the LA area than it was 500 years ago. Now, what species live here are vastly alien to what was here 500 years ago. There are still species that were here pre-European contact, but there's a lot of stuff that was brought in from over the world. So a lot of introduced species because we also happen to be a very busy port. And then also because of human modification for things like making the LA River a year-round river, it allows for species that normally would not live here thrive, like bullfrogs, for example. Bullfrogs, yeah. Where? There was always wetlands, but it was but there was it went up and down. It went, yeah. It just wasn't dry. Yeah, I guess it wouldn't ever totally dry up, but it was a very strongly seasonal river. You probably heard the statistic about 80% of the flow in the LA River is treated municipal discharge, which is like the fancy name for sewage. Although from an ecosystem point of view, this now like completely changes it from like an intermittent river to like, oh, now it's just a year-round river. So like it causes like for example, like frogs that were adapted to this intermittent system that like now get displaced by bullfrogs because they can't outcompete them. But this makes this whole what's known as urban ecology really fascinating. To me at least, and then other people, because look, I have citations, is that humans are ecosystem engineers, like beavers are building dams and everything like that. And so to sort of paraphrase Stuart Brand of the Whole Earth Catalog, if we are to be as gods, we might as well get good at it. We make these environments, other things live in it. How do we make the best of this? Cities are not apart from nature. They are just another ecotope essentially at this point. And we are predominantly an urban species, so we really kind of have to figure out how to make the most of this. I think we passed that threshold like only in the last few years, but we are now a majority urban species. All right. So to spearhead this, we looked at the Los Angeles watershed as a way of studying this environment because and I'll get into more like why, but like LA is everything literally drains in the watershed. So if you're looking for biological signals, you essentially are going to use the watershed and you're going to collect everything across this urban area and see like what ends up in the water. And it's a good way of getting sort of the biological signals. Plus, I mean, like river systems are a big driver of any ecosystem's health. So there's this really large project that was funded by Metabolic Studios. So this is Lauren Bond, and her whole setup is her whole studio, and they fund art and science projects. It's funded sort of this preliminary study, and there's a whole bunch of partners involved in this, and it became this Protecting Our River project. So these are like all the partners. So like I've worked with all these people here. So I'm directly affiliated with like Cal eDNA, but I've worked with all these groups in one way or another. Oh yeah, Squirt. Okay, look at all those logos. It's like scientific NASCAR. All right. So this initial study, so we did 3 time points. This is in sort of the peak of the COVID shutdown, so 2020, 21. We did 3 sampling rounds a few months apart because we wanted to get dry and rainy season. 12 locations in this case. And we did collection of sediment on the riverbanks. We also collected filtered water. And then we did this, it was fall of 2020 and then through until the spring of 2021. And so these are all the sites we collected at to get sort of this broad overview of what's in the river. In terms of like, how do we figure out what's in the river? We use this really cool technology called eDNA. What is eDNA? Wow, good question, me. All right. So eDNA is a technique that's become feasible, really practically feasible in the last 10 years because sequencing and computational power has gotten so cheap. But here's a really good picture of this. I didn't draw this. I wish I did. But step one is, and this, there are some people in the room that have done this, is collect the actual environmental samples. So in this case, this should look really familiar. If this is filtering a water sample. We've done a ton of these. So you take this, you take water, you force it through a filter, and then you collect all the biological material in the filter. And then there's this huge, there's like this like scientific easy bake oven process of like getting from like whatever gunk that is to like extracting the DNA and to get it into fragments. And then what you're doing is then you have like all these DNA fragments, you clip out certain chunks of the DNA that you want to sequence. Then you do something called PCR polymerase chain reaction, which is just like you keep photocopying and duplicating DNA millions of times. Now you have enough DNA to easily sequence it. You run it through a sequencer. If you have any questions like how the sequencing works, it's pretty, I think it's fascinating. You sequence all of this, you're reading everything at once, and then it becomes data at that point. And life, just like with Taco Bell, only has a handful of ingredients. We only have to read 4 letters. And then we just, from all those letters, we have these little fragments, and then there's a lot of computational power we have behind this, but we can identify from those fragments what species they are likely to belong to. And so you'll end up with literally billions of these little fragments floating around, and from it you can go like, oh, that's from this carp, or that's from a coyote, or that's from like this water plant. And then from there, then it becomes like reading the tea leaves, so to speak, and to do the ecological analysis. But there's actually like well-designed scientific kits now that can take from the mud, from like with a filtered water, extract the DNA, sequence it, and now like, okay, this is where it becomes like a computer science kind of problem. All right, any questions on this stuff yet? Stunned silence. Okay. All right, so just going a little bit more detail here, like what this is, we're using a technique known as metabarcoding. There are a lot of techniques to deal with environmental DNA. The one we use, which is a workhorse of environmental DNA now, we look for what are known as particular like metabarcoding regions, so specific genes, and we don't actually sequence the whole gene, but what we're doing is we're looking for genes that are shared across lots of different species. So the reason why you want to do this is you want to find something where if you sequence this particular gene, you can get like a broad swath of like the tree of life. So an example is like genes that code for ribosomal DNA. Basically, ribosomes are needed to make proteins. Everything makes proteins. So like people will look for like ribosomal genes. So things are known like as one for bacteria is called 16S. There's another one for like multicellular organisms known as 18S. There's a lot of different genes that are specific to different gene regions. And we don't sequence the whole thing. We're actually sequencing like subsets of that. So like a couple hundred base pairs, a couple hundred letters of DNA at a time. And what we're actually looking for are what are known as, trying to sequence what are known as hypervariable regions. So like if you see this thing where it says V1, V2, and so on, those are known as hypervariable regions. So if you're sequencing like a gene region, you want to sequence regions that evolve relatively rapidly so that each species will actually look different. And you want to get a couple of these what are called conserved regions in between. And the conserved regions, the way to think about this are coding for very, very key processes in like, let's say metabolism or protein synthesis, that if you make any mistake, it probably kills the organism. So those tend to evolve very slowly. Whereas the ones that, whereas the hypervariable regions are things like you can get away with like more mutations and it won't kill the organism. So if you look at evolution across like just zooming out, if you look at whole genomes, the rates of evolution at different chunks of the genome vary drastically. Because some things are just super important, you never want to mess with it. And some things, you can just kind of mess with the code and it doesn't matter. Like lots of software is like this too. Like you don't want to mess with the kernel. Okay. So this is what we're doing is we're looking for particular like regions. So this, like we actually design certain DNA sequences that clip onto the left and right sides of the particular gene region we want to look at, snip it out of the DNA, then just double it a bajillion times with PCR. And then that's actually what we sequence. So that's metabarcoding. All right, that summarizes about a year of grad school for me. Okay. Hope this is illuminating. Okay. So this is a bit technical here, but what for our particular purposes, if there's not usually, when people do metabarcoding, they usually don't use what are called like one primer pair, like one of those like gene regions they want to clip out. They usually want to use multiple ones because there's you want to get a lot of redundancy. So the way to think about this is that when I talked about, there are gene regions that are hypervariable that can evolve really quickly, this isn't universal. So like one of the things that you'll find out is that lots of species may look almost identical depending on which gene regions you're like sequencing. So they are called like what geneticists refer to as degenerate. So the idea is that a lot of these things will look identical, but if you look at a different gene region, they'll actually look distinct. And so what you want to do to hedge your bets is you want to use a lot of gene regions and sequence those in parallel. And you do this because some things may look identical on one gene region, but it'll look really different on another. And so you'll pick up like different types of diversity across it. So you're trying to get like a broad swath of the genomes of species to get a more complete picture. By the way, like eventually what we're going towards as computational power even gets more ridiculously cheap is something called metagenomics, where we're not going to be sequencing gene regions. We're literally going to sequence everything. And at that point, then it becomes this a bigger problem of managing all that data. But now you're not looking for particular gene regions. You're literally like getting just everything and reconstructing genomes computationally. That's a talk for a different day. But the algorithms behind that are the same algorithms we actually use to reconstruct the shredded documents from the East German Stasi after the fall of the Berlin Wall. Anyway, so we do all this. We use 6 different primer sets, and these are to target different broad categories of organisms. So 16S is something that codes for ribosomes for bacteria and archaea. Archaea are like another type of single cell organism. Then there's a broad one for eukaryotes, which are you and me. And then we have another one that's also for eukaryotes, so it's CO1, which for those who know is cytochrome oxidase 1. And then we have ones just for fungi, ones for plants, and ones really specifically just for vertebrates. All right, we're going to get all these. We get all these gene fragments. We get every sample we collect, and then what we want to do for some quality control is we only keep samples if we get at least 2,000 sequences. Because if you can't amplify anything, you probably assume it's like statistically it's just noise. And we only want to say an organism's there if we get at least 2 copies of the sequence. Again, it's just to make sure that we're not just getting a fluke. So, all right, quality control. Okay. And, sorry. All right, so once we got all these DNA fragments, how do we know what species they belong to? And I'm going to do my best to summarize somebody's project that she's worked on for 5 years. So there's a postdoc at UC Berkeley I work with named Lenore Pipes, who's a genius. And I'm not saying that I'm not saying that lightly. But she developed this piece of software with a PI, Rasmus Nielsen, known as Tronco, which by the way means tree in Chamorro from Guam. And what Tronco does, and I'm going to do my best to try to like summarize this algorithm, is think about when I talk about you take those primer pairs where you have that sequence that's like from the left and right. If you're looking at this DNA, so you have like a primer sequence, like left and right. Now, what her software does is it does a digital version of this where it says, okay, let's take the database of everything that's ever been sequenced. So you can get that online. So like there's repositories that are like where every time like a genome or some like environmental sample sequence and people know what it is, we actually store it on a database and say like, hey, this gene fragment belongs to like a cheetah or something like that. And it gets stored. And what her software does is it does a digital version where it says, okay, I'm going to take like, if you give me a primer sequence, like to the left and right, I'm going to run this across every gene, like I'm going to run this across every genetic sequence in that database. And I'm going to do a digital PCR. I'm going to clip out all these little, like what are called amplicons, these little marker regions. And I'm going to do that for a given primer pair. And I'm going to do this like it's like literally billions of things online. And it just like runs across that. So now you have like a list of like all these little like gene fragments and like what we know it is. So like species level or genus level. So you have like billions of these things now. So you do all that, you generate all those fragments. Now you have this huge table that is, hey, this fragment probably belongs to this kind of algae. And if you were to if you were to try to then use like, if you take someone's data and then like use those gene fragments to like taxonomy tables to look up and try to place what's what, you can do it, but from a computational science point of view, that takes forever because every time you have to look across the whole table and say like, hey, What is it? And you scan through like, 100 billion rows. So for those who do a lot of programming, this is something that scales way more than like n. So this becomes very inefficient very quickly. But this is a method that people used to do because we were not dealing with nearly as much data, so it didn't matter as much. Now we're in this realm of like hundreds of billions of fragments. This just does not become practical anymore. So what she did is develop software that does this digital version of PCR, builds that table, and then organizes it by creating what's known as a phylogenetic tree. So it takes all those fragments and it says, hey, sequences that look similar to each other, it'll say like, these are probably two species in the same genus, and it'll make them as like adjacent branches. And there's like a whole like probabilistic way to do this, but like she's literally taking all those fragments and like assembling a tree out of it. Why does this matter? It matters because when you make it a tree, now instead of having to scan across 100 billion rows, now you only have to zoom in on 10,000 because now you know, oh, only go to this branch. So she's taking like all these fragments, making a huge tree out of it. And now when we take our data with all the DNA stuff that we don't know what it is, it only has to go across like one little part of that tree at a time. And so it greatly speeds it up. So instead of things like literally taking months, we can now do it in like a day or less. And then it also does like this whole probabilistic measurement, because with sequencing, by the way, you can get errors, there's uncertainties when you're reading it. Sometimes we get what are known as ambiguities, if we don't really know if it's an A or a G, for example, but we just say it's in between. There's a lot of things you can do to say like, and this is why this is all color coded, where you go like, I may not know what that is to species level, but I might know what it is to genus or family. Or if somebody hasn't seen this before, but it looks similar enough to something else we've sequenced, we may say like, oh, I know what phylum it is at least. Which is really important when you're dealing especially with like fungal and bacterial sequences, because those are things where we just have like big gaps in our knowledge. Oh, come on in. Oh. So we have like big gaps in our knowledge. So we don't know, because especially bacterial diversity is like 99% of all biodiversity on Earth is that. So like, anyway, this is like our best guess. So sometimes we don't know yet, but we can get like a decent job of placing it. All right. So now we can take like all this like DNA that we get from the river, we can sequence it, we can tell you what it probably is. Now what? So now you've literally just generated like just this dump truck of data. I mean, like we're dealing with like hundreds of gigs up to terabytes at a time. So with eDNA, now we have like the what of biodiversity. What lives here, what showed up, in the last, couple of days or whatever. Now it gets into this whole exploration of like, what's the why? What's driving it? What's the puppet master ecologically? So like, what's driving like why this species shows up or not? And this is like where you get into like, now you have all this data and like, how do you do anything with it? Well, the answer, of course, is more data. All right. So this is where we get into now with remote sensing, which is we want to get environmental data. So what we're doing is we're tying together environmental data with biodiversity data. So like, we want to be able to say like, hey, I don't know, like all these species show up these particular places in time. Usually it has something to do with a couple things. One is species to species interactions, and the other one is interactions with the environment. So in order to start addressing these questions, you have to get environmental data. And some of that you get in the field, and we do that. And people do that with all sorts of projects. But if you want to start doing this at scale and you want to do this efficiently, this is where you start going to a remote sensing. That is like satellites are constantly scanning, recording all sorts of information. So this is an example of what's known as the Sentinel-2 satellite system. Sentinel-2 is used for a lot of things that are really pertinent for it's near infrared to near ultraviolet, and it gets 12 different bands. And you can use that information to look at things like ocean color, which is really useful for like looking at algal blooms, cloud cover. You can infer a lot of things with photosynthetic activity by looking at like red and near infrared. So like fun fact, chlorophyll strongly absorbs red but reflects in near infrared. So if you look at those two channels, you can actually get a good proxy for photosynthetic activity. So anyway, you get all this satellite data. So this is like something now that we're starting to tie in with all this biodiversity data to see like, hey, what's influencing this? All right. So where do we get all this data? So I'd mentioned Sentinel-2, which is a big satellite system, but we have a lot of different map layers that we're actually pulling, and they're all curated by Google Earth Engine. So Google Earth Engine is a big repository for a lot of different public data sets that are updated from satellite metrics, from a lot of different groups like EPA, CAL FIRE, Like there's another one that does like literally nothing but light pollution called VIRS, VIIRS, which was originally developed to look for things like give you proxies for like urban activity just from like artificial light at night and gas flares. Habitat quality maps that were, that are generated by like combining other data sets. Anyway, so Google Earth Engine stores all this, and I worked with so we have an engineer on a team that I work with. We put together this whole chunk of software that takes all the locations when people collect samples, and we record the lat and long and the date. That goes to Google Earth Engine and say, hey, extract all the environmental data from these sets. And we literally, I think for our project, we're getting somewhere on the order of 250 different maps that we're extracting. So both like social variables, so things like demographic data, but also environmental metrics. And so you end up with these, like, when you get all this data, you end up with like digitally, it's a version of a map stack. Like aligning and stacking all this stuff is its own fun project that I spent a year and a half learning how to do in grad school. So we have something like, hundreds of layers, things like elevation, exposure to light pollution, lots of different bioclimatic variables like rainfall, average annual temperature, variability. And the map layers vary a lot in resolution. Some are 3 meters. There's other layers out there that we'll probably get in the future from NASA. They're doing this whole, like, what are called Earth cubes that are developing that are going to be on the scale of centimeters potentially, which is like really crazy high resolution stuff. If you want to be really spooked, I'm sure the NSA like works on a lot of stuff that's like what are sort of things that are beyond like the normal index of refraction, where you can probably get down to centimeter resolution stuff already, but it's not civilian yet. So have a nice day. Anyway, so the resolution on stuff is like that we're dealing with is 3 meters to 1 kilometer. So the 1 kilometer stuff is like broad things like long-term climatic variables. 3 meters are things like we can get for elevation actually is really, really high resolution because we have really high resolution radar that just scans and does that. So we get all that, we stack all that information, we extract all that information in these pixel stacks, like when and where we got it. So now you have like, hey, all these species showed up, and this is like what the elevation was, this is what the pH was, this is how bright it was, all that associated with every sample. Cool. So you extract all that, and here's an example of this is a paper that's in review that I've been working on with some other folks. This is a paper actually like looking at the Woolsey fire back in 2018. And so if you look at these little circles here, what we did is we extract all these pixels inside each circle. This is sort of like an illustration of what we were also doing in the LA watershed. But this is like an example of elevation extracted from there. And this is the data associated with each one of those like coordinates. We're doing the same thing hundreds of times for like each location. So we have this really rich data set. So we're doing this similar technique with some other projects with DNA. In this case, where this is all drainage from the Woolsey fire, which is a talk for a future day. All right. So we got biodiversity, environmental data, scientific peanut butter and jelly. This is so great. All right. What are we going to do? So this is where machine learning comes in. And I hate using that term, but that's what it gets referred to as. So we're doing machine learning using physical measures of the environment and biodiversity to start to figure out like what's going on ecologically, because this is like the only way we know to really manage data sets they're just insanely huge. Of course, what's step 2? So how do we actually do the machine learning or the modeling part of this? So this is where I'm going to get into. I'm going to talk about one technique. There are lots of different techniques, but I'm using one that's pretty robust, and it's actually I like using it, 1, because it's fairly accurate, and 2, because it's kind of how humans make decisions. So, and it's known as random forest. So I'm going to tie this all together, but random forest, the idea is if you're dealing with these really complex multivariate systems where you just want to go like, hey, how do I know, given like all this data, like that this is like something is going to show up, for example, like what my output state is going to be. You make what are known as decision trees. You take your data set, you randomly take some fraction of it. Usually people do like 80% of your data, and you'll have, let's say, dozens of variables. And you make a tree that branches at each unique variable. So like it could be in our case, it would be a variable that like has like every time like, so you have everything branches at elevation, then you have ones that branch at rainfall, and then branches at how bright it is at night, and so on. And what you're doing is you're taking all your data and said like, okay, I want to predict whether or not some species shows up. And you make this giant decision tree that says, okay, here's what the data was when that species showed up or it didn't show up. And you just go like, was the elevation above or below this? And you make a fork in the tree. and you go like, all right, what was the temperature? Was it above or below this? And you fork the tree. And at the end of it, you go like, did the species show up or did it not? And you make these trees with, you build it with like, let's say some fraction of your data, and you say like, hey, did it show up or did it not show up? And you randomly like take your data like 80% of the time, you do this, you build a tree. And then you end up making like an average decision tree after doing this like thousands of times or maybe a dozen times. You average the results together, so you have like literally one big decision tree, so that if you give it like, here's where all my variables are in my data set, if it was this bright, if it was this, this level of salinity and so on, or if it's this much photosynthetic activity, that this species is going to show up based on this decision tree. So this is a commonly used technique in machine learning because it's like really like straightforward to implement, and you can scale up to like really crazy levels. There are a lot of other ones that I've also used in ecology, but I really like using this one. I'm going to stop here because I'm assuming someone might have a question.
On the previous slide was Japan. Is that a Japanese model, or what was the Japan and US only on the previous slide?
Wait, what?
Right there. In the modeling, it's like Japan, US only, US with Japan for.
Oh God, I don't know. I just did a screen grab. Sorry.
Oh.
Yeah. I was just trying to I was doing an image search. Okay. Yeah. But yeah. So this is like a toy example of it, like where you're making forks and variables to predict like, hey, what's the outcome? In this case, I guess it's apple. All right. But this is used a lot in all sorts of things, not just ecology. But there are a lot of different machine learning techniques. I like using this one. Okay. So.
You have a question from the audience.
Oh, she asked to repeat it.
Repeat the question. Oh, the question was. No. We're all gonna listen to your shame. Okay. The question was, if you look at this, like this part here that says modeling, and says Japan, US only, and US with Japan prior. I have no idea what that is from. I was just doing an image search, and it was not on Pinterest. Thank you. Okay. All right. So decision trees. By the way, this is a lot how human beings make choices. Oh yeah, Glenn, what's up?
No, tried to hit the emoji thing.
What was the final decision that you want to make by using these trees?
Okay. I'm going to get to that in a minute. Yeah. So good question. I'm just about to get to it. So how are what are we applying these random forest decision trees to? So our inputs are all these map stacks. So it can be like what watershed this happened to be in, slope, soil type, land usage categories, and so on. So just picture like you have a location where you say like, hey, at this coordinate, these are like all the environmental conditions, and I happen to see this hawk. And you'll have lots of, and then what you do is you would then look at locations where you have environmental data, but you don't have any observations of, let's say, a Cooper's hawk. And what you do is you build decision trees that say like, under these conditions, I saw it, under these conditions, I didn't. And what you're doing is then building an aggregate decision tree that says, okay, under these conditions, I'm probably going to see this hawk show up or not. So you end up building a unique decision trees particular to each species in this case. And it's going to be based on like the environmental data you have, like, and where you have your observations. Now, the next question I'm probably going to get is, well, how do you know where you didn't see this? And the answer is this wonderful scientific fudge factor we do called pseudo absences. Because most time in ecology, you don't have what is known as true absence data. Usually you don't have data where you say, I'm looking for this species and then I didn't see it. Usually you have data where you just say like, hey, I saw this species, and that's it. You have what's known as presence only. So to get around that, you assume that you make what are called pseudo absences, where you just randomly generate locations on your map, extract the environmental data at those locations. And where places are where you don't have data, you just say like, I'm going to assume most of the time that hawk didn't show up. Those are pseudo absences, or sometimes known as background points. And it's a way of helping train the data set. Now, the answer, like the question is probably going to be like, well, how do you know it's not really there? I'm like, look, something Bayesian statistics to first order. Anyway, but most of the time you can assume like it's probably not there. And then at least where you have what's known as presence data, you have true presence data, and you do a reasonably good job of training your model. Now, this gets into like, how do we evaluate model accuracy and all that sort of stuff, which I'm going to get to in a minute. But that's kind of how we train that. I don't know if that answers your question, though. No answer.
Yes, it does. Thank you. Thank you.
Yes. All right, cool. So anyway, that's how it works. So you have to use what's called presence only data in this case. Now, once in a while, somebody does an ecological study where they really have true absence data. That is rare. Primarily when I've ever worked with stuff like with my data or other people's data, it's usually presence only. The other thing that kind of helps is if you're dealing with large volumes of data that you're collecting, is you can usually assume like a lot of the errors kind of smooth out sort of in the statistics. That kind of helps as well. All right, yeah.
Question.
Yeah.
So what happens if you have an invasive species? Does that count as errors if you have a large amount of data? And how do you decide that's a worthy error or not?
Please repeat the question.
Okay, so the question was is like, if we have, let me see if I'm paraphrasing this correctly. Is like if you have an invasive species, how do you know that's an error or not, right?
Yeah, because you wouldn't have data on that prior, right? So wouldn't you ignore that as noise?
So in our case, and actually a lot of ecological case studies, we don't care if the species is quote unquote invasive or not. We're literally taking data on like the species. Whether or not it's invasive is irrelevant for our data. So we're just treating the data as like, I don't care if the species when it was introduced 500 years ago or if it was here natively. We're just going to get like, did the species show up here at this time, and what were the environmental conditions associated with it? And then we evaluate each species individually for this model.
Just whatever it is now.
Whatever it is now, yeah.
Were you asking about like a brand new invasive?
Yeah, like what happens if it's brand new though? You are correct in the sense of like if it was 500 years, yeah.
Oh, yeah,
right? Like earthworms, who cares, right?
Yeah.
But what happens if it's brand new? Do you categorize that as just like noise?
Okay. So following up on that is what if it's a very recently introduced species? By the way, the term invasive gets very complicated for ecologists, and I can get into the mud slinging on that one. But any sort of introduced species, even if very recent, so this becomes the trick. There's a lot of data filtering that you can get into with modeling like this. One of the things is you can say like, I'm only gonna let's say use observations like within this time bracket. I'm also going to try to do what's known as spatial thinning, where if I have like a lot of observations nearby each other, I'm going to assume a lot of them are redundant observations and throw them out. There's also what are known as spatial biasing that you can try to adjust for, which is not so much with eDNA, but a lot of like when people take photographs and upload that data, is that you end up with a lot of observation hotspots. So for example, in LA, Griffith Park is a biodiversity hotspot. If you look at raw observations, that's completely erroneous just because everybody likes taking photos of birds in Griffith Park. So you can compensate for that by like building models of like how things tend to cluster in certain areas and then compensate. But again, it's like, does a recently introduced species influence where other species show up? Yes. And that gets into a more model that I'm not going to get into yet, called a joint species distribution model, where you're not only doing a random you're looking at. And the next question is, how computationally intensive is that? And the answer is insane. There's a reason why we don't do that a lot yet, because take what you have and then add in like a couple zeros to the computational resources.
Right. To the nx power.
We're going to get there. I mean, in 10 years, I'll probably be a more common technique. And like, but we kind of do this because it's a first order approximation that we can do relatively efficiently. Yeah.
How much does the data lag for all the places you're getting data at Cal Fire and whatnot?
Yeah.
Do you wait a month behind, or does it take 2 months to process and then predict? Or like, what's from when the data is gathered to when you're making like predictions off of it? How much is the lag time from the date of it being gathered and kind of like the output?
That is an excellent question. This was a lot of time we spent like when we were coding the environmental extractor that we were doing to like, okay, go grab those values. We are assuming a lead time of a month. Well, so we'll get like, because like a lot of those satellite layers are dynamically like updated. And what we'll do is we'll just like look at environmental data like in the month leading up to like when the sample was collected, and that's it. So some of the map layers that we have are like long-term layers. Like there's some layers like the climatic variables are like 30 years at 30-year averages. But for things that are dynamic like Sentinel-2 or VIIRS, these are just like a month. Yeah. So, and this will actually show up in some of the modeling I'm going to show from like our data analysis. Yeah. Yeah. Oh, okay. Yeah. So these are excellent questions, by the way. This is pretty much, I'm assuming the reviewers are going to come back to us on anyway. Oh, raise hand. Wait. Oh, Glenn, what's up?
Suppose the Cooper's hog poops in the river. I collected my samples from the river. It can go miles. How are you going to get environmental data on him when his poop went miles away?
Oh, man. Okay. You're going to love our Woolsey Fire paper, because that's literally all this is like dealing with, is that here's what we found out. And there was a Nature paper that just came out 2 years ago. Watersheds essentially are just giant nets of biodiversity for eDNA data. Everything in that watershed will get concentrated in the river. So the way to look at it is so what you end up finding out is your environmental signals that are relevant are only relevant for, if you were to like picture that whole watershed, look at all the environmental conditions upstream of like where you collected that DNA. That's what's going to drive what you see, because everything's draining into that very point. So when you're bringing up like that Cooper's hawk poops in the river, it's just like, where do you get the DNA? And this is like one of the best and worst thing about eDNA is it is frustratingly sensitive. It is astoundingly sensitive. So like we'll pick up signals, you know, that are like tens of kilometers upstream. So you end up having a picture, if you're doing like aquatic sampling, essentially assume that everything upstream in that watershed is like aggregating the environmental signal. So you can't say like at the very immediate point that that's probably the most relevant data set. It turns out if you look at environmental conditions on average and that watershed upstream of you, that's probably what's really driving what you're seeing.
What about like wind, like the same end is really crazy.
So the other question is, what about wind? If you're, for our kind of data, not so much, because we're getting stuff that's like in the riverbeds itself. But wind transport, or like a scientist love to use aeolian transport, can be a driver for stuff if you're dealing with like loose, dried, like topsoil stuff, or like dealing with like, or DNA that clings to sand particles or dust particles. And that can be transported long distances. And that becomes especially relevant as people start to do more what's known as like taking air columns, passing through a filter and doing eDNA sequencing off the captured air filter. So the answer is like, yeah, we're probably going to get some of the stuff that's windborne, but the concentration of DNA in sediment and water is like a factor of 1,000 times greater than what you're getting in air. So it's getting swamped, so to speak. You're going to get some of that, but it's going to be swamped compared to like what we're seeing in water. But that whole thing about like, am I just getting DNA from bird poop? I'm actually, Glenn, I can tell you right now, like, so for another project we're working on, we were doing some sampling in the Sacramento Delta. We were finding all sorts of primate DNA, like chimpanzees, bonobos, orangutans. And it turns out it's because there's a primate research facility upstream of where we were sampling, and not like just up the road. It was 50 kilometers upstream. So
wow.
eDNA is like insanely sensitive. So like, yeah, we've also had other instances. We were doing desert hot spring sampling, and some of our volunteers we're doing work at the Desert Hot Springs Zoo, or the Palm Desert Zoo. We were getting giraffe DNA showing up in desert oases in Palm Springs.
Then
right.
The tree, the data that you give to the trees, the random forest model, it won't know. It won't know whether the Cooper's hawk, it will just know that it was in this watershed. It won't know its local environmental conditions.
Yeah. So this is actually like what we're getting to, and this is like one of the things in this other paper I'm working on, looking at eDNA and a different system from like drainage from the Woolsey fire, is that the local environmental conditions, if you're dealing with stuff you're collecting in riverbeds, the local stuff like right by where you collected is not the driving signal. It's looking at everything upstream in the watershed. But we're collecting that data anyway. So, but yeah, it turns out if you're dealing with like riverborne stuff, yeah, the watershed is essentially acting as the aggregator. Now, if you're looking at just topsoil that's not like draining into a riverbed, then stuff that's like immediately there, those environmental conditions actually are the more relevant ones. So that's one of the big things we're finding out with like eDNA is like it depends on the material you're extracting it from. If it's waterborne, it's just whatever's draining into it. If it's soil, then you can deal with like the local environmental conditions. And if it's windborne, we don't quite know yet. That's more of a more bleeding edge technology. Yeah. All right. These are awesome questions, by the way.
So the LA Zoo is upriver from where we were.
The LA Zoo is upriver from us, and so like, yeah, we can. So the LA Zoo is upstream from some of our sites. So the nice thing is like we know what species are in the zoo. So like if we see elephant, we're going to assume that it's the LA Zoo. This is, yeah, there was like, again, with the airborne DNA stuff, there's a proof of concept. This is in Copenhagen, but this group did air filtering and sequenced just when they pulled from the air in the Copenhagen Zoo. They were able to detect every species in the zoo.
Wow.
Yeah. So now this is only metabarcoding, so it's only at species level. But once we get to metagenomic stuff and you can look at individual markers, then you can eventually do like individual human detection from air column stuff. So anyway, just thought I'd throw that out there. You want to be completely paranoid. All right. Yeah. Yep. All right. So this kind of gets more into like that's like training this, but your random forest, this is like, again, you're doing 80% of your data, you build a tree, then you randomly take another 80%, you build a tree. And then what you're doing is when you do your 80% and you build that tree, you want to then take that 80% tree that you did, and then you want to test against the other 20% of your data you didn't use to build the model to see how well could I predict the other 20% observations that I saw. And you do this to evaluate the model. In our case, for the data we got from the first 3 rounds of the LA River, so the first 3 rounds we've completely processed bioinformatically. Then the subsequent rounds we're still in the process of doing all the sequencing. The first 3 rounds from those 12 locations, we have that data. And this is what we were doing is we're running this through like, the thousands of species we detected. And it is thousands. We did an 80-20 split for every species. And we did this 10 times for every species and aggregated the tree. And we did it in the 80-20 split is to do the training and testing. And then you can do this to actually evaluate this, which is model accuracy, because then people are asking like, great, you build a model. How do you have any idea how accurate this thing is? And you can do model accuracy with random forest, which is another really easily, which is one of the reasons I like it. And what you're doing is when you build your model and you say like, hey, how often does my model predict something's there and it is? That's a true positive. How often do I predict that it's there and it isn't? Which is, so you're dealing with like, and then how often do I predict something's not there and it isn't? So that's a true negative. And what you're trying to do is you want to get your true positive and true negatives. And then you also want to get your false positives and false negatives. So how often do I say something is there and it isn't? How often do I say something isn't there and it is? And so you end up building this binary table that's like, how often can I correctly predict what I'm seeing and how often am I incorrect? So when I'm like using my 80% to predict the other 20%. And if you take your true positive rate plus true negative rate minus 1, like you do, you end up with something called the true skill statistic, which is a model accuracy value. And it goes from 0 to 1. Typically, anything above 0.4 is considered accurate. That is like your decision tree is a good predictor of like what you're seeing. So like if you have your like whatever 300 variables, you make a tree with it. How accurately can you predict that this species shows up, so to speak? We used a 0.5 indicator threshold to say like, hey, if my true positive rate plus true negative rate minus 1 is greater than 0.5, I'm going to say that that particular species can be accurately predicted given the environmental conditions we have on hand. So now that's not to say that we couldn't predict it if we had other types of data, but at least for the data we were collecting from all those satellite photos and everything, at least we can do that. So this is how we evaluate the accuracy of the models. All right. By the way, this is a map of Finland. Okay. And someone's asking me like what the training and testing set is here. I don't know. Again, it's Google image search minus Pinterest. Okay. So you can do model accuracy. And here's the other cool thing that you can do with random forest model evaluation is you can do what's called evaluating variable importance. So remember, like I said, like you have hundreds of variables in your decision tree. Your 300 variables are probably not equally important. In fact, most of them are probably redundant. For example, like if you have a variable that's like human population density and elevation, they're probably going to be very strongly collinear because people are not going to tend to like live up on like slopes of mountainsides. There's lots of things like that, or like artificial light at night and population density are usually strongly correlated, although not always. And then like, so anyway, you have a lot of collinearity and you have a lot of variables are probably not that important. And so in random forest, the way you can evaluate model importance is kind of cool, is you just randomly just like start ripping variables out of your decision tree and then see like how well your model, like how well does your model still perform if you just like randomly like remove elevation, let's say, or randomly remove temperature. And if your model still works, like almost the same as it did before, then that variable probably is not very important. So you can actually calculate what's known as the variable, like the relative importance of, or rank importance of those variables. So what you end up finding is like you'll have 300 variables, but probably like 6 of them are doing most of like the driving of that variation. So this is a way of reducing your model complexity. So you don't have to say like, oh, if I have these 300 parameters, I know what I'm doing. Because to quote Enrico Fermi, with 5 data points, I can, with 4 data points, I can fit an elephant. With 5, I can make it move its tail.
Do you weight them or do you just rank them?
Okay, so the question is, do I weight them or do I rank them? These are they're essentially weighted by like doing the determining the ranking, but there's no additional. So what you're talking about, like with that, there are other models that people do that to like try to like add additional weighting factors, and that gets into more other types of machine learning techniques like neural networks. I'm not doing neural networks. Yeah. Anyway, like I'm just talking about like sort of like the one technique we use for this paper, but like there are lots of machine learning techniques that do get into that. This is just unweighted, just randomly start pulling wires out of the model, and if it totally breaks everything, then that was probably a really important thing. And that's how this is done. So we can take this, reduce the complexity, because usually like most species, it's like probably driven by like a handful of factors, but they're not always the same factors. So we do this for every species individually. All right, what did we learn today? Okay, so I'm going to summarize what we found out from those 3 time points in 12 locations across the LA watershed, which is like, I don't know how many thousands of species at this point. I think it was something on the order of like 12,000, in case anybody really wants to know. But of all those, we found 374 potential indicator species. And by indicator, I mean those that have who we can predict with a model accuracy with a greater than 0.5. That is, with the environmental data we have on hand, that those species can be reliably predicted by the environmental conditions. So that's what we're treating as indicator species. By the way, like indicator species are a really key concept in ecology. Like they're telling you something about the environment. We're not assuming anything. We're literally using machine learning to like bootstrap and figure out like which ones they are, which is especially important for things that you can't take photos of that people aren't studying classically, like bacteria. And what we found out of all those bajillion species, the ones that look like they're potential indicators, environmental indicators, 58 of them are prokaryotes, 316 of them are amateur or eukaryotes. And there also turns out to be very little overlap if you look at each time point. So over like those 3 seasons we sampled, the type of indicator species that show up in the winter are not the same that show up in the spring. And one of the reasons is probably because the LA River has a very strong seasonal flow. We have a very punctuated rainy season. So what is a good indicator of the environment when it's raining heavily is probably not a good indicator like, this time of year. Which is like one of those like well duh kind of things. But a lot of times in science, it's nice to find things that are like results that are boringly reassuring. Because 90% of the time you find out like, yeah, okay, I kind of figured that, but at least your model's not telling you something totally insane. So it's saying like, okay, things change a lot in the river. It's a strongly seasonal river. In terms of like the environmental drivers, like what's really important for like what shows up or what doesn't, the variables that seem to matter the most are things we can gather from the Sentinel-2 satellite, which are things that are updated like every 4 days. So these are measures of things like photosynthetic activity, cloud cover. So things that like vary very quickly. The things that matter that are less important are more like human impact variables. So like land cover, like how much impervious surface, soil conditions, whether it's like just there's dirt, whether it's plant cover. And then the ones that seem to matter the least are the long-term climatic variables. So things like 30-year average temperature, for example, doesn't seem to matter that much for like what predicts what shows up in the river. So that's the sort of, I'm summarizing a ton here, but that's what we found out. And if you want to see some really fancy charts, I spent days and days and days coding. Oh, there they are. Oh man, I'm so proud of this chart. You have no idea how much code it was behind this figure. Okay, so this is the rank importance. So the variables that are the higher number here are more important. The variables that, and this is like round 1, 2, and 3. These are the seasons, the sampling seasons. And if you look at these, the distribution of values, the variables that are significantly different between seasons are labeled in black. The ones that are insignificant, like the ones that pretty much don't seem to change in importance between seasons, are highlighted in red. But these are like the shorthand names for these variables. But to give you a rough idea of like what's going on here, the ones that matter the most, the highest rank importance here, these are these variables that say things like B1, 10, and so on. These are the Sentinel-2 bands. So these are things like, okay, like what's the brightness in this color band? And these are variables that are driving things related to like photosynthetic activity, things that evolve very quickly. And we get like 4-day average data on this. So they update very quickly. And this is using this is a CO1 primer, which is like sort of a broad, like multicellular organism, like swath that we're looking at. The environmental variables that seem to matter less are these class here, actually, well, the least. These are the long-term climatic variables. So bio 1 is like average annual temperature. Bio 12 is like average annual rainfall. Anyway, these are like 30-year averages. These aren't that important. Then the ones that are kind of in between are all these variables that deal with things like human impact. And then some other stuff like the slope doesn't seem to matter that much, but like things like human impact seem to matter, not nearly as much as like stuff that like again, like photosynthetic activity that evolves very quickly, but like it is important. But it's kind of like it's all over the place. All right, great. So now we have all these indicator species, potential indicator species. They change really strongly over time. And the stuff that like environmental conditions that change really quickly seem to be more important than things that are long-term averages. Great. So we just like literally summarize like hundreds and hundreds of gigs of data to tell you something that like you could probably guess beforehand. So what do we do with this? All right. Well, this has been, it's kind of, we found from this that like, okay, like just from those 3 time points are these like strongly seasonal patterns. And so one of the things now we wanted to do was go out and collect data over a longer period of time, so a couple of years, to look at, well, how much of this is due to seasonal influence and how much of these are longer term changes like over multiple years. So like, because especially because you have longer term climatic changes, like how much of this is just because like, oh, it happened to rain that year a lot, and how much of it is just like you have a gradual shift in the climate. So this is why we started doing a longer term study. We are now looking at 4 locations. This is a joint project between CrashSpace and UC Santa Cruz. It's funded by the Los Angeles County Board of Supervisors. Thank you, funders. And we did 11 time points. So we have like a much richer data set now. So this is 11. And now we're looking at this question of seasonality. And this is also just like a proof of concept to do long-term eDNA monitoring. This is more novel with eDNA projects. So a lot of eDNA projects are more opportunistic. They're not like, we're doing a long-term study repeatedly at the same location. So that's relatively novel. So hopefully we're going to set up stuff that's like beyond the scope of this grant. But anyway, this was a start. This also kind of gets into this issue. And like, I've worked on a number of eDNA projects, is you're kind of reinventing the wheel with a lot of these projects over and over again. So a lot of these tools are not quite standardized. A lot of the methods aren't standardized. And so this kind of leads to this whole thing, because I've worked on a lot of these projects, and a lot of my colleagues have, is can we sort of make a set of standards so that there's a lot of more interoperability between eDNA data sets? Because one of the things in ecology, and we've done this now with like, if you've ever used iNaturalist, or eBird or any of that stuff, is like all that data actually gets aggregated in a big standard database known as the Global Biodiversity Information Facility, or GBIF, which allows for like literally like thousands of projects to like all be integrated in the same data set. That means other people can come along and query across huge amounts of data to do their own analysis, myself included. So we're hoping to make something like that, but for eDNA data. So take like stuff you can take photos of, tack on 2 zeros to the species list, because it starts getting really nuts. So this was kind of like, all right, let's see, can we start standardizing and make like a commonly used data platform? That being said, this always runs into the risk of the XKCD. There are now 15 standards, but we're at least attempting. So to that end, about 2 years ago, my colleagues and I, we got a grant. This is from UC Santa Cruz. We got a grant to actually build the web platform for people to take eDNA data and location data, and like we'll automate the whole process of like, if you give us your DNA data and like when and where you got it, we'll automatically do all the environmental extraction for you. We'll automatically do all like what species is it, and store all of it in a database, and you can publish it and share with other people so that like you can start aggregating data across multiple projects. This has been insane. I've been working on this now for 2 years. I have now become more software developer than human at this point. So anyway, yeah, I'm barely an ecologist anymore. I'm just coding. So like, this is the platform built. It actually exists now. I was running stress tests on it last night. So this is kind of where it's all leading to. So this is like screenshots, like what does some of this data look like in our platform? So eDNA Explorer, which is going to be the focus of the next talk, but I'm going to give some introduction on this. We can actually store all your sequence data, do all the taxonomic assignment with Tronco. That includes all the common names. And if you want to know like how many months of my life I spent just to get the common names and these little icons, you can ask me later. Somebody here in the audience actually helped me with that. Thank you. And we also do a lot of comparisons, like, because this is probably like coming back to like, well, the Cooper's hawk poop cherry, was it really there? We actually do a lot of comparison now between like, hey, I saw this with eDNA. Did someone take a photo of this in this area? Is this thing really here? Or is this just like an erroneous thing because somebody like got elephant poop on their shoes and then like did some volunteer work the next day? So we actually can automatically compare eDNA results to photographic results. So it's traditional observations. We do all the environmental metadata extraction, everything. And so like we end up running all this stuff. We say like, hey, each sample, what percent of your samples does this thing show up in? And like every species, or if you want to look to genus or whatever, all automatically calculated for you. There's a monstrosity of like bash code, R, C, Python, all the languages, everything except Pascal and Julia, I think. All right. Anyway, so this is like more stuff you can do with it. We can actually generate maps to say like, hey, where did I see with eDNA? Where do people see with it in person? Where did they see it over time? Yeah, PostgreSQL for the win. All right. And then we can automatically do all the map extractions for you. So you don't have to write your own code and go to Google Earth Engine. Yeah. So anyway, thanks, Google. So yeah, all sorts of stuff. We got all these layers, all of the data. And we're also building in some like all the collaboration tools so that other people, you can basically make a profile pic like you can on, well, it's like LinkedIn for Science. God, I hate LinkedIn. But like, anyway, so like LinkedIn for Science, but we have that so you can like find collaborators, see what other people are working on, or find out if people are good at X, dealing with ecology. So we can actually help speed up a lot of these collaborations and whatnot. Because this is like of interest, not just people doing ecology, but precision agriculture, all sorts of stuff that involves anything with biodiversity data. So yeah, very proud of this. I am absolutely exhausted by it. Anyway, and here's where, this is stolen from our pitch deck. We're taking all three networks of life, people, and methods, and synergy. Wow, look at that. They all merged together. Give us money. Okay, so this is like, this is actually like working on turning this into a B Corp, by the way. The B is for good. Okay, so this is where we're coming to with this. The LA watershed was kind of a great test case and motivation. And this is actually our the LA watershed project is the flagship project on eDNA Explorer now. Those of you who are in person and actually want to see what some of that data looks like on eDNA Explorer, you're welcome to. And with this, I'd like to thank all these people that help out. So people that help collect the data, some are in the audience right now. Also with my colleagues at UC Santa Cruz, Professor Rachel Meyer, who worked with on getting the grant for eDNA Explorer. Postdoc Chloe Orland, who actually I worked with to get the grant to fund the follow-up sampling, and Ajith, who is a lab tech who has done a lot of the lab sampling, but also has helped a lot with like coordinating fieldwork in LA. Same with Max Usuda, who's also helped me a lot with computational stuff. Protect our river, LA County Board of Supervisors, helping pay for all the subsequent sampling, Crash Space, and all the community scientists of LA. Yay. Anyway, all right. With that, questions? I got the good one so far. Ooh.
Go ahead.
Where? Oh, okay. Someone clapped. Okay. Oh yeah, Glenn, what's up?
Oh, could you go back to the slide with the one that you were proud of with the different I forgot what you call them. It was a bunch of stripes, bunch of little things, little shapes on the graph, and it showed that weather things that were seen by the Sentinel satellite were the most important. Yeah, that one.
Yeah.
What are the most important human things that were even more important than some of the Sentinel things?
Oh, okay. So the ones that significantly that deal with like that deal with human activity, because a lot of these ones that are like intermediate, the ones that humans have like the most influence on are what's land cover. So like basically like is it covered with grass? Is it covered with trees? Is it covered with like, yeah, because that's going to influence a lot of things. Like does like water infiltrate into the soil or just like run straight off in the ocean? Or like what lives, I mean, it's going to influence like a local biodiversity and drainage. So like the land cover one is like, that's it.
It's just that. Oh, wow. Okay.
Yeah.
They don't really matter. That's good to know. I mean, that's good
Well,
news.
Well, okay, so before I get to saying like, wow, that's great, I'm just going to throw this Pepsi bottle in the river. Before we get to, it's more of like, remember, like I'm only looking at this recently. This isn't like a 100-year data set. So remember, we're looking at this at this ecosystem in this like incredibly altered state. So in terms of like, if we were to look at this over 100 years, a lot of human activity is going to shoot up in importance. But this is like, what this is saying is like the day-to-day stuff, because we're looking at these like short-term, like this is like months at a time, because it's already so altered. This is now quote unquote its new natural state. So like this is like, so remember, like it is an ecosystem, it's a very rich one, but it is insanely altered from what it was 500 years ago. And so like day-to-day stuff, like, yeah, we're not influencing that much day-to-day, but that's because we've already like influenced it heavily now over the last 100 years. And then, you know, I would argue with humans were ecosystem engineers for thousands of years before, but like the last 100 years have been very intense. So to really get at that, you'd have to like start looking at what's known as ancient DNA, don't worry, and or paleo DNA, which there's the lab. We actually work a lot with a lab at UC Santa Cruz that does ancient DNA. You'd want to look at things like pollen core data and like old DNA going back hundreds and thousands of years to see like where you look at proxies for environmental change and then proxies for change in biodiversity. And that's where you see like humans have been a big influence, especially as the ice ages ended. There is another recent paper in Science that basically looked at the last 15,000 years and like looking at controlled burns and human activity. And basically like humans have like influenced a lot of ecosystems with controlled burns over like the last 15,000 years.
Usually census data could overlay that.
You can use
when a population came.
Yeah, population is going to give you, but that's such a coarse aggregate. So you'd want to try to get like more like fine grain, like actual direct chemical measurements of the environment. Like you'd actually start looking at like paleo, like you'd actually look at isotopic data. Yeah.
So the difference between like Arroyo Seco and the LA River, because one is a natural system,
yeah,
and one is on a concrete channel. So that's what you're talking about with like land.
Oh, so that's
so you had them mark the box on the thing.
Which
the eDNA collection form, there's a box you can mark.
Oh yeah. That's where I'm marking all the substrate stuff. Yeah. So like the question is like, you know, with the land cover is like is that distinguishing things like Arroyo Seco from the rest of the LA River? Because Arroyo Seco is not concretized. That's an example of it. Yeah. So like impervious surface is like a land cover that has a huge influence. Yeah.
No, my question was why are some of the bars on it kind of forked as compared to the other ones, which are more like a flat thing?
Forked. Sorry.
Like the shapes on the actual graph.
You're talking about like that one right
now? So these are So these charts here. So this is showing like, so each like variation or that swath like for each variable, it's showing the variation in the samples. So for example, like because this is like over 100 samples, so there's a lot of variation. Let's say in substrate with that. So it's showing you like the basically the standard deviation. So yeah, so like some of these vary a lot, and then where it's significantly different between each sampling time point, that's when it gets color coded as. If it's significantly different, it's color coded black between seasons. But yeah, that's showing you the variation. Some of them just don't like. There's not that much variation here. So like Bio 14, which is related to rainfall, just does not vary that much year to year in LA. So anyway, that one's like barely shows up at all. But then some of them like have huge variation across our data. So substrate, which is like dealing with like soil types, that varies a lot. Yeah. All right. Other questions on this? Oh yeah. What's that?
So is who owns an website, and was that what you got funded? Is that like the kind of like the end a deliverable, or like what's the ongoing plan for that?
So the ongoing so eDNA Explorer is so that's funded under, so it's currently funded under a foundation. Last 2 years, that funding is we're getting additional funding and working on like spinning up as a B Corp. But like it's that's separate than this, but it's like but that helped motivate this. Because I'm working with like an eDNA lab in Santa Cruz, and we do a lot of eDNA projects all over the world. So like, and then we collaborate with a lot of people who do stuff all over the world. So like a lot of work in, let's say, Rwanda, Copenhagen, South Africa.
So it'll be around.
Yeah. So this is like this project, we can find like additional funding and personnel, like keep long-term monitoring the watershed, it'll happen. But this is all getting like aggregated into eDNA Explorer. Yeah. Yeah.
Quick question. So I know a lot of your slides were talking about using machine learning,
Yeah.
using AI or whatever. But how do you verify that those tools that you use that are clearly automated are working the way you want them to? Like, who verifies the tool itself? Do you have a way, or do you just kind of just blindly accept like that's just how it is, and we trust it enough, or like who, you know?
What so the question is like, how do you know that any of these tools we're developing work? So that's basically just becomes a push and pull between like going out there and collecting the data. And like, I mean, like in that, so, but I mean, do you have like a specific part of that you're thinking of?
One of your professors, you said, had made her own tool in the AI, right?
So Tronco. Yeah. So how do we know how well Tronco,
like, how do you even say like, oh, it's like a 0.5% differential or whatever? Like, how do you even?
Do you mean like having up the software?
Exactly.
Yeah.
Like, how do you be confident about it?
I think it's just mathematical, though, right?
So there's going to be error bars and everything. So what you're doing is you're saying like, so the question is like, let's say with like Tronco, so like how well do I know that the taxonomy of all these DNA fragments? There's a lot of things you just have to factor in and say like, okay, I have like X% uncertainty with like was each base pair sequenced correctly. So that gets into the model. And then like you have other models where you're predicting like it, you have that. You also have other things where you just say like, hey, I've never seen this sequence before, but it looks this similar to something we do know. And like you have a model that says like, well, then it's probably like in the same genus as this, but it's not the same species. But at the end of the day, like this just becomes, that's just the whole scientific process. Like you'll build it and you may find out like, yeah, you predicted stuff that turns out not to be correct. And you're going to have to end up tweaking the model. And that's the case with everything in research.
Right.
Yeah.
So when you write a paper,
yeah,
you know, on the bottom you talk about potential errors, right?
Yeah.
Do you bring that up at all?
Oh yeah.
Or do you like peer review of like the code?
Yeah.
Oh yeah. So the code is like, the code's going through peer review. Yeah. So like, and it'll just subsequently, there's going to be other stuff because then someone's going to come along and probably try to develop another tool and say like, you have the following flaws and this improves upon it. And that's just the way it goes. Like,
open source.
Yeah. So like the code's open source. So like, and that's the other thing too, like with peer review, like that's kind of like the whole thing with science is like, you don't say like, this is true. You just say like, here's the uncertainty. This is the level of uncertainty I'm willing to live with. And like, given the best guesses I have, this is what it is, which makes science hard and totally antithetical to how humans think, which is why we didn't invent it really until 350 years ago, because we're not evolved for science. We're just not. No, we're not. I mean, we're just like, the human brain is not wired for that. It's not wired for calculus.
Going back to social science
Yeah.
experiments and like realizing like the way they set their data and the way they like, for the social ones that are more subjective.
Yeah.
They're looking back now, like if we were rerunning the study, like we would have gotten different results, or we think it was like, you know, biased in this way because they were in social science, they were like making conclusions about humans in like the 60s. And we're like, oh, if we were to do that now, I don't know that this would be such a strong causation or correlation.
Yeah, with social sciences, the best description I ever heard with most social sciences is like, have you ever heard the acronym WEIRD?
Yeah,
white, educated,
Yeah.
industrialized. Yeah.
Yeah.
So like, so basically, like, what do sociologists like? What do they usually find? Like, the readily available study subjects are undergraduates that have to also be on campus. And like, the best analogy I ever heard is like, that would be like only studying penguins and saying, I know birds.
Yeah.
You probably know penguins really well, but you probably don't know birds very well. Yeah.
The crazy thing is like a lot of books
Yeah.
after that were based like this
Oh,
is
yeah,
fact, you know?
Yeah.
Yeah.
Well, we heavily studied like one segment of humanity. Like maybe some of that stuff's universal, but we don't know. Like, but yeah, we have like a wider swath of data here, but like this is always getting improved upon. Like, and as we go towards other methods, like what are known as metagenomics. So now you're not just like trying to find gene markers. Then you start getting this, like, as the computational power gets more, as computational power increases, you can actually just get all the gene fragments and statistically assemble like the genomes of everything you find. So like now you can actually start disentangling individuals. You can start like, you know, but that's a more complex problem. Yeah. It also allows you to get viral DNA or viral sequences. So in case anybody's asking. All right.
Looks like you've chats. I don't know if that's
I-
if you check those.
Oh yeah, chat. I don't know.
Let's
Like
see.
the bubbles blue.
The bubble's blue. Yeah, you're right.
Let's see what we got.
Nope. All right. Goodbye, blue bubble.
Mhm.
All right.
I have a question.
Yeah.
So was there anything that you found that was really unexpected besides the primate research?
Anything we found was really unexpected.
In the watershed?
We didn't find we didn't find anything like that was like really crazy. I mean, it was things like, okay, like what are like really strong environmental indicators, and they're just like, for the most part, they're all like these bacteria that like don't have any common names. They're all like some sort of oxyphotobacteria. So they're like some like photosynthesizing bacteria apparently is like a really good environmental indicator of river chemistry. And it's like not anything majestic like a heron. It's just like most of the things that are really good indicators are like all the bacterial stuff. But like to me, that's actually not shocking because it's the bacteria and the fungal stuff that are like your sort of direct interface between the abiotic and biotic worlds. And like they're really going to be driving your biogeochemical cycles. I mean, to picture it, like the true biodiversity of Earth and for almost all its history is all unicellular. The quote Stephen Jay Gould, this is and always will be the age of bacteria. You know, like, and so like those are where all your strong ecological signals come from, are like where are there fungal hyphae, where are there bacteria doing sulfate metabolism? Yeah.
Wasn't there more eukaryotes than prokaryotes in the thing?
Yeah, there were.
Like
Yeah.
nematodes or something?
So basically almost all the eukaryotes are all, we're all like either things like fungi, like, or really small, like, or even unicellular eukaryotes.
Not even just like nematodes, like giardias.
Yeah, so it's all the single stuff. Yeah, all the single cell stuff is the strongest signal. But think about it, like those are the things that have very rapid generation times, and they're going to be responding very quickly to environmental changes, and they're the ones driving all the chemical changes that drive the ecosystem. Like
bigger things, more generalists, no? Like
bigger things are just, well, their reproduction time's slower, so that there's a lagging response. And like they're not the ones that are doing the more rapid fire changes of like, like producing hydrogen sulfide or like metabolizing methane or all that sort of stuff.
Well, we'll see after Taco Bell.
We'll see after Taco Bell, that's right. But yeah, like the ecosystems are all driven by bacterial stuff. Like right now, half of the oxygen you breathe is cyanobacteria. And the crazy thing about like now is half is a cyanobacteria, but half of that cyanobacteria is literally like one super stripped down species known as Pelagibacter ubique. It's the most common organism on the planet, and is a super simple cyanobacteria, has an incredibly tiny genome, and one of every 4 breaths you take is just driven by that little cyanobacteria.
Nice.
Yeah, it's called Pelagibacter ubique. It also has it's the name like SAR 11, which is like Special Adaptive Radiative Clade number 11 for short.
That was your first guess.
Yeah, I know, exactly.
Could you then sort of say from this that if we create, like, as we move forward to try to fix everything and make the city healthier and more better for biodiversity and all that, that we could actually focus on the conditions that are most beneficial to those indicator species of healthiness?
Yes.
Like when I do gardens, like we start with soil health.
Yeah, so
that is the basis for the whole garden.
Yeah, so like, I mean, no, like what? So the question is like, if you want to try to do like biological remediation, like, do you just want to focus on the unicellular stuff? The answer is like generally, and like for soil stuff, it's the microbiome of the soil and the fungi.
Right.
Yeah, and like so, and
fix that first.
Yeah.
So instead of worrying about like protecting a single bird species,
Right.
like you could actually think about like what makes the whole city happier, like makes that bacteria happy or that fungal species more dominant in your city.
Yeah.
Then you fix the whole ecosystem.
Yeah, you don't go for like the charismatic megafauna. If you want to save the ecosystem or keep it functional, you save like it's the things you can't see. It's all the bacterial and fungal stuff. And that's actually what we found with like the Woolsey fire stuff with ecosystem recovery. The stuff that comes back first is fungi. They're the ones that send all the hyphae back into all that supercharged soil immediately.
Right.
And they're the ones that drive the creation of soil from mineral. Yeah. So like, and that was a case, a billion years ago. It's the case today. Like, I mean, so now people don't ooh and ah and take photos of lichens, but that's so, well, some people do.
I remember Seattle.
Oh yeah. And lichens, by the way, are amazing. They are symbiote. Mushrooms are pretty too, yeah. But like, yeah, but the charismatic megafauna, the things you can take the big photos of and are on National Geographic, like they can be important species, like, famously, like starfish are keystone species in intertidal tide pool ecosystems. But a lot of stuff that really matters is just super tiny. Like I said, like the most common, like 1 every 4 breaths. This is like super stripped down cyanobacteria that floats in the ocean.
Right. So if we're going to design like a pocket park in a city, and we're going to make it a biodiversity like supportive element, or we're doing a biodiversity corridor, then we focus on what will allow the fungal network to work through that whole corridor, and that will create a biodiversity rich corridor, instead of just like planting the right kind of flower.
Yeah, support the bacteria and fungi. I mean, it's like, I hate to be this reductive, but it's a microbiome thing. It's just like yogurt works for people, and effectively it's what we're finding out with like ecosystem remediation is like you're applying like yogurt on a big scale. Delicious yogurt. Yeah. I mean,
This is super random, stupid question, but like, I don't know if there's animal shelters near the LA River, but I've wondered if anyone's ever, because animals will get caught locally at the time from being in the area. I wonder if there's any samples or things, because I know like also like a lot of germs pass throughout the whole shelter, but if there's any studies that have been done on like the local shelters, like what kind of things are coming out of them, or even just the waste product, or just how that's correlated, because those are wild animals that would be impacting local areas.
Animal shelters I don't know about, but like I mean, just but there's like anything with like animals in general. Yeah. So like, fun fact. So like, about Toxoplasmosis gondii?
Yeah.
That's from cats.
Yeah.
So basically, everywhere you see humans, you see cats, and you also tend to see like Toxoplasmosis gondii. And we see this actually like in coastal environments with like lots of humans. You see like lots of Toxoplasmosis gondii showing up in shellfish because they're filter feeders.
Oh, and people put it in the toilet and stuff.
Yeah. So like, I mean, and it's like also because diseases jump back and forth. I mean, like we just went through an epidemic, so like diseases jump back and forth between humans and other species all the time. Like the 1918 flu probably started with cattle in 1916 and jumped back and forth to humans, and like then exploded as the 1918 influenza epidemic. So like, you'll see a lot of that. Some of this is like the more you know, the more you probably don't want to know. But like, because you also end up starting worrying about things that like, does it really matter, or is it probably just sort itself out? 99% of the time, it sorts itself out. So you just end up freaking yourself out. Just assume you're awash in a sea of bacteria. There's as many bacterial cells in and on you as there are of you. You are a colonial organism. You are an island in a sea of filth. And like and for 80+% of Earth's history, all life was unicellular. Multicellularity doesn't even start to take off until a little more than 500 million years ago, which is practically yesterday. Whereas life itself is at least over 4 billion years old. So you throw a dartboard anywhere in the timeline of Earth's history, most of the time you're going to get pond scum. And we're recent. We're really recent. And it just shows, because like you're just crawling all this, and you actually would drop dead if you didn't have like all these bacteria in and on you.
The other things is we're seeing people with cats and dogs, their kids grow up with less allergies because they're bringing in soil into the house that they wouldn't normally be exposed to, because we're so. There
seems to be something that with like just exposure bacteria and immune system stuff, but it's like it's still up for debate, because it's very it's like, if I say it's complicated, but like there's probably something to it, I would venture. Like these are very very multi-dimensional systems, and we have now methods of tackling this complexity, but like it gets really crazy. Like this is like, I spent a lot of my life hunched over a Unix terminal, like trying to get computers to do what I want them to do, and unfortunately they do exactly what I tell them to do, which is usually not what I want them to do. So computers do exactly what you tell them to do, unfortunately. Remember, the computer didn't screw up. It did exactly what you told it to do. The problem is that you don't know what you told it to.
Yeah, it's kind of like your earlier question. You're like, if it's working right.
It's more like the key thing with science, and what is so hard about it, is it is only that which is falsifiable. If you can't falsify it, it ain't science. Don't bother with it. But all right. I guess on that note, science something ego death. All right. Thank you for all coming to the talk.